Great. Well, thank you all for coming today. I'm really excited to have a couple experts from a company called Velsera on the call with us today. We have Andy Bredemeyer and Gargi Nanda, and they're both from this company, which has kind of a unique infrastructure and way of dealing with variant interpretation and reporting workflows. I think that a while ago, a couple years ago, we had Period DX on the call, and recently Velsera and Period DX kind of joined forces. I'm sure Andy or Gargi can speak to that just a little bit. Um, but thank you so much for being with us today and sharing your expertise with us. Really, continued discussion in this field really helps us move forward in the field. So thank you so much. And with that, I'll turn it over to you two.
Beth, thanks so much. Really appreciate the introduction, and we're excited to tell you today about Velsera's efforts to build and maintain a clinical knowledge base that clinical labs can use in conjunction with our software platform to interpret and sign out reports, clinical NGS reports in oncology quickly, right, with high quality. That's the goal. Is there's a lot of information out there associating biomarkers, genomic biomarkers to patient management decisions, and it's a lot to keep track of, and we endeavor to make it easier. So I'm joined by my colleague Gargi Nanda, who's in Pune, India, visiting the office there today from Mumbai. I'm in St. Louis, and I'm looking forward to sharing with you some of our work and a little bit about our platform as well and how users interact with our knowledge base there. So you set me up pretty well, Beth. So indeed, you may not have heard of Velsera. You may have heard of Purion or Purion DX. Purion and Seven Bridges and Ugentech are three companies that came together to form Velsera earlier this year. So Seven Bridges platform is a research and discovery platform that is actually a key part of another GA4GH driver project, which is called the NHLBI Biodata Catalyst. So this platform supports a lot of researchers managing lots of data sets, doing secondary analysis or building their own workflows to automate, as well as doing cohort analysis and other sort of discovery efforts. Ugentech's Fast Finder is a lab workflow orchestration solution that has a lot of adoption in PCR-based lab workflows, and in fact, a lot of adoption during COVID for automating workflows and qPCR analyses for COVID testing. Purion's Clinical Genomics Workspace is our flagship product that we'll talk about some today, but mostly about the knowledge base. So Purion's been around, or Purion was around for about 8, maybe 9 years before becoming Velsera. Our platform is used by many labs internationally in order to interpret and sign out cases, both germline, inherited disease testing, and in oncology. We also have a lot of services we provide to help labs stand up their tests, help them validate, help them with a reimbursement strategy, as well as things like assisting with interpretation and sign out if bandwidth is not available at a lab. So we'll be talking today about former Purion's solution, the Clinical Genomics Workspace, and the knowledge base. The Clinical Genomics Workspace is really our web-based platform. It's an all-in-one solution from analysis through to reporting for various disease indications and assays, really NGS support. So this starts with identifying the important variants that have some patient management impact, associating that impact and being able to communicate that to an ordering physician and a patient on a report. And it goes beyond just that interpretation with taking raw data in for secondary analysis all the way through to reporting. We've really built integrated seamless workflows for multiple different, many of the available commercially commercially available assays in oncology and in inherited disease, and are really agnostic to technology with respect to sequencing and capture and assay kits that we have built integrated solutions for. We'll focus on these first, left two most components of what one can do in the clinical genomics workspace, where really we'll describe this core knowledge-based component of our solution, which really describes the conditions under which certain associations, be they prognostic, therapeutic, or diagnostic, should be made, not just to those commonly described alterations, but it also makes intelligent predictions about rare or novel variants based on their characteristics when they share characteristics with those alterations that are more commonly identified and have clear associations. So we'll delve into that some. So here's our agenda. I'll be doing an overview of the knowledge base for the first 15 or so minutes here. And I'll hand it over to Gargi to talk about how we perform verification and validation of our knowledge base and some benchmarking studies we've performed as well. And then I'll give you a few minutes on how a user experiences the knowledge that we've curated in our knowledge base in our web-based platform, the Clinical Genomics Workspace. I think we're probably going to hold questions to the end. Feel free to put them in chat, or if you can't wait, raise your hand and we'll try to call on you, but we may hold mostly till the end and address them.
That sounds good. I'll keep an eye on that.
Thanks, Beth. All right, so this core of our solution, the clinical knowledge base, is really based off of clinical evidence sources, drug labels from drug agencies, the EMA and FDA, clinical practice guidelines in the US, NCCN and ASCO, as well as the ESMO guidelines in the EU, and then published results of human studies, the results of trials that have been peer-reviewed and published that have some conclusions to draw from their study. These are this is emerging evidence on new biomarkers or new associations to existing known biomarkers that have not yet been adopted by practice guidelines or don't have an approved drug, as well as clinical trials. So a key part of successfully curating all of this knowledge in a way that's useful to a lab is we've built a series of ontologies that we rely on to make these connections, that we cite these sources for, as well as some others listed here. This includes drug ontologies, disease ontologies, and a biomarker ontology. We've built a sort of library of biomarkers where we've associated biomarkers or that are described with words in these documents and associated them to discrete conditions under which or discrete conditions that describe the type of variants or specific variants that qualify as that biomarker. And so in this way, we can make a quick association between some statement in a drug label with a whole set of variants or conditions that describe a set of variants, where the FDA intends for those variants to be an indication for this drug, for instance. And so we use some software-aided processes for searching, extracting, and then largely manually screening this information from these sources, and leveraging also some other public databases shown at the right. We curate discrete associations between biomarker and a drug response or non-response, prognostic value or diagnostic value from each of these documents, as well as clinical trials. And we perform this on a weekly basis. So we're looking for changes to these documents, new versions, new publications, or say trials that have just been published, or rather been added to registries like clinicaltrials.gov and EUCTR, or they've changed their recruitment status. So we're doing this update weekly, and it's all of the processes I describe here are done under controlled SOPs that are compliant with ISO 13485 medical device manufacturing standards. So we've really developed a set of really rigorous processes to make this so these curation steps reproducible and yielding high quality final product. Now, from here, we have a bunch of discrete associations we've made from the sources that are described at top. And we then take a step to aggregate these into sort of assertions that represent consensus. Okay, sometimes there's a lot of if not duplicate, at least agreement between these sources, or sometimes some especially published evidence may not agree, and so we sift through that, and we'll spend some more time in a few minutes looking at how we manage that from the PubMed literature and look to build consensus and express assertions that are reliable there. And we perform this on a weekly basis as well, and we also write interpretive statements, expert interpretations about these assertions that we aggregate that can be used in our system. And then finally, on a monthly basis, we perform a formal verification and validation of the knowledge base. So here we're performing testing to make sure that the knowledge base meets our specifications, as well as testing the accuracy of the content, both assertions from these sources and clinical trial matches, based on orthogonal sources or orthogonal opinion. And so only after we perform this rigorous formal verification do we make a monthly knowledge base release. Now, this knowledge base is used in the clinical genomics workspace alongside the discrete associations from these various sources and trials here, as well as public databases that are commonly used to identify common polymorphisms or other information from these sources. And then finally, something that I believe Pirion pioneered and is now relatively common among other vendors, but we have a sort of a shared interpretation network where interpretations that are written and signed out by laboratories on our system are then made available to the rest of the network, so they can read others' opinions or look at their peers, and either use some of their interpretive text or modify it as they wish. So all of this comes together to serve as a knowledge base in the CGW. The verified curated knowledge base is also part of medical device solutions, including Illumina's TSO comprehensive assay that's alive on the EU market today. Good. So our knowledge base is really designed to aggregate the knowledge to create ready interpretations that especially labs across the spectrum, from academic through to nascent commercial labs can benefit from. I think I've hit on some of these points, but just to reiterate that we're constantly curating from practice guidelines and drug labels, so all oncology guideline information from US and EU sources are incorporated into our knowledge base within weeks of publication. We've curated all relevant oncology trials associated with biomarkers from the EU and US sources, have scoured PubMed literature from the past 10 or so years, and have covered all genes that are relevant for solid and hematologic tumors, at least based on assays that are available today in the market. Just a quick point here to illustrate how much knowledge is being generated out there. Our knowledge base, here this example is our solid tumor knowledge base, over an 11-month period grew by about a third. So the y-axis here represents knowledge base rules. This is a rule is a statement of logical statement of conditions under which content should match, describe the genomic characteristics of an alteration. And you can see much of this growth has come from the published literature, from our PubMed curation, but some of it is also from increased recommendations or drug label approvals from NCCN, FDA, et cetera. We think this is illustrates the importance of having a curated source from which you can readily identify information for reporting. In fact, it ought to be automatically applied such that you don't need to perform any of your own searches or otherwise try to identify which alterations are meaningful for reporting. I'll focus for a few minutes on a couple of strategies I think this group might find interesting that we employ in our knowledge base curation and maintenance process. The first is to delve a little bit more into the data model that we employ to apply patient management, associations to alterations in a flexible and powerful way. Our metadata model lets us describe a broad range of various conditions that we might want to use to describe what ought to match to content in the knowledge base. So we describe this sometimes as these conditional if-then statements. If a certain condition is met, then this particular association of drug responsiveness, prognosis, or diagnostic value can be made according to various sources listed here in these examples. So this could be an HGBS C dot, P dot, or G dot syntax, but the simplest example shown here is just at the codon level, which is actually a categorical variation in a sense. We're saying in AML, if there's a codon R132 mutation, which could be a whole bunch of DNA level changes, right, that are going to make a single amino acid substitution, that is associated with possible benefit from ivosidenib in AML. And so an example might be an R132C alteration, but you could change this to, H, or any change here according to our knowledge base would then trigger this association. If you step out further, you can imagine now at the exon or domain level, we can specify a range of genomic or protein codon coordinates, within which certain knowledge should apply. And in this case, we're looking at an in-frame alteration within exon 20, with an exception. This one won't qualify under this knowledge-based rule, but any such in-frame EGFR exon 20 insertion in NSCLC is associated with potential benefit from this drug, according to several both drug agencies and the NCCN. So here, a duplication, an insertion, any in-frame insertion would qualify. We can also describe protein consequences that qualify. So in this case, in ovarian cancer, we can describe that from codon 1 to 1854 within BRCA1, any truncating alteration or a frame shift that ends in a truncation or any overt nonsense alteration is associated with likely responsiveness to PARP inhibitors. And here we've defined the end of codon 1854 because there's evidence that truncating alterations at the very C terminus of BRCA1 protein are they're not prone to or not subject to nonsense mediated decay or otherwise maintain their function. And there's actually benign alterations in ClinVar, as many of you may know, at the very C terminus in those genes. So I won't go through all the examples, but structural changes, fusions, copy changes are all supported by the model, as are co-mutations. So in this case, an HERX loss and a mutation in IDH2, both being found in the same tumor, have a unique meaning with diagnostic vasculocytoma, distinct from the finding of these alterations independently. So we can express that, as well as conditions for clinical trial matching that may be negative. If you have an exclusion criterion for a trial that's a particular alteration, we can describe that in the negative as well, so that we avoid matching even if the other conditions are met. Genomic signatures like TMB and MSI are also supported. So we find this is a powerful way to be able to describe the conditions as that are described in sources that we curate, where there's a biomarker named, and sometimes it's really not explicit what that is. We define what that is, and we can even describe it in a way that alterations that are rare or novel that are definitely would qualify are also picked up automatically by the system. And then about the aggregation that we perform, this becomes really valuable when it comes to emerging evidence that's published in the literature, but is not yet picked up by a practice guideline as a recommendation. AMP-ASCO-CAP gives criteria for what qualifies as a tier 1B or a tier 2C finding, but it's not real explicit. And what qualifies as consensus, what's a large study, what's a smaller study, and it's also difficult to do consistently. And so we've developed an approach here to define specific thresholds and aggregate the individual papers' evidence to determine what qualifies as a 1B or 2C biomarker. So just to step through the sources here, at guideline and drug label, there's less discrepancies and there's less debate about those. Occasionally, the guidelines will make statements about options, treatment possibilities that are not exactly recommendations, so we treat those differently. There are large published studies that either have a favorable drug benefit or prognosis, or unfavorable, just a thumbs down, or sometimes there's some trend, but there's not a clear, maybe statistically significant conclusion that the authors can state. And then there are smaller studies, likewise, that may have clear outcomes, clear associations, or less clear. So when we look at all this together, clearly when there is guideline and drug label evidence, which is usually, there's good agreement across these sources, this qualifies as tier 1A on AMP-ASCO-CAP. When it comes to larger studies, if we've essentially described conditions or rules, an algorithm about what sort of consensus we need. How many disagreeing papers can we put up with? How many do we need that are in the same direction in order to call that consensus in the field at tier 1B? So here's an example where most of the evidence is in one direction and there's enough in that direction to qualify. And likewise for tier 2C, a similar threshold has been defined, though it differs between therapeutic and prognostic based on what the AMP-ASCO-CAP guide joint recommendations state. And so here, smaller studies suffice, and fewer studies for consensus. So we've felt that this is a useful model to follow to try to routinize, standardize the way that aggregated evidence is looked at for tiering on a clinical genomics report. I'm going to hand it to Gargi next to describe some of our testing approaches for validation and verification of the KB.
Thanks, Andy. For the next 5 slides, I'll delve into our routine verification, validation, and benchmarking processes for our knowledge-based content. We'll explore the various quality assurance methodologies we use to ensure accuracy and reliability. These approaches are crucial in guaranteeing the accuracy, integrity, and comprehensiveness of the information within the knowledge base. We assess the knowledge base for variant classification performance by comparing the classification assignment of the IVD knowledge base to medical director assigned variant classifications obtained through finalized reports in the Clinical Genomics Workspace, or CGW, Clinical Data Repository. Our second approach is to test the sensitivity and accuracy of reporting clinical associations using orthogonal testing. Orthogonal database testing utilizes independent databases that are assumed to represent the ground truth with respect to clinical assertions, example, therapeutic, prognostic, and diagnostic claims, and/or classifications. While this method has limitations, it allows for objective calculations of true positive concordance within the knowledge base. These metrics help track the overall performance of the KB and pinpoint areas that need improvement, enhancing its quality. Our next approach banks on knowledge-based expert panel or KEP reviews. So the knowledge-based expert panel focuses on consensus building strategies for handling ambiguous high importance biomarkers within the knowledge base. The aim is to align these strategies with the best clinical practices available. This process ensures the KB maintains accuracy and relevance in addressing challenging biomarker scenarios. In this slide, I'll be talking about a couple of these approaches. So in the first approach, we assessed the accuracy of Velsera KB for assigning clinical relevance to variants as compared to medical directors. As per AMP-ASCO-CAP joint recommendations, clinically significant variants are classified as tier 1 or tier 2, and variants that lack clinical significance are classified as tier 3 or 4. We studied how well the Velsera KB identifies the clinically significant variants compared to what medical directors in the CGW agrees upon. We looked at 8,834 cases from 34 organizations. and checked close to 6 million combinations of genetic changes and diseases in solid tumors using NGS panels. We checked how accurate, specific, and sensitive Velsera KB is. Out of the 6 million variant disease combinations, close to 49,000 combinations gained more than 66% agreement on clinical relevance assignment across medical directors within a lab as well as across labs. Velsera KB matched the lab's judgment on whether these changes were clinically relevant in signed out cases. It agreed on 113 clinically relevant and 49,000 clinically non-relevant ones. However, there were 6 cases where it disagreed. These differences were because the KB might have correctly identified emerging biomarkers or when the medical directors relied on old evidence. Also, sometimes the medical directors overclassified changes that are common in the population. Through this approach, this next approach, we wanted to see how well Velsera KB matches high confidence claims across solid tumors found in clinical interpretation of variants in cancer or VICC database. This helps us understand how sensitive the KB is in reporting claims about treatment, prognosis, and diagnosis for specific combinations of diseases and genetic changes. We used real-world genetic variations from Velsera Clinical Genomics workspace to represent the same biomarkers mentioned in VICC's claim. These genetic variant disease pairings were examined within the Velsera KB to identify overlaps between the assertions in VICC and the information present in the KB. For instance, let's take the example of an assertion from VICC stating the NRAS mutations in colorectal cancer that do not respond to panitumumab. In CGW, we identified 2 real-world combinations relevant to this biomarker disease pairing, NRAS A146V in colorectal cancer and NRAS Q61R in colorectal cancer. These were then queried within the Velsera KB to assess the concordance between the VICC assertion and the information present in the KB. Here we see 2 overlapping sets of assertions from Velsera KB that correlate with the VICC claim. So in total, we tested 8,589 different connections between these real-world genetic variations and 200 strong claims from VICC. These claims covered 33 genes and 38 types of solid tumors. What we discovered was that most of the VICC claims, about 78%, were supported by the Velsera knowledge base. However, the remaining 28% lacked sufficient recent evidence or consensus in the field. Some of these claims in VICC focused on single genetic changes, while Velsera KB linked those genetic changes happening together. You might remember from a couple of slides earlier where Andy talked about our strategy to curate co-occurring genetic instances. Now, if you look at the graph on the right side of the slide, you can see that we could only check 5% of Velsera KB's claim using the available information from VICC, as shown in the orange fraction. This shows us the importance of using more sources to accurately measure and compare the KB's performance. In the final slide from this section, I want to talk about the approach we periodically use to evaluate the accuracy of Velsera KB in correctly classifying benign or likely benign variants in ClinVar as tier 3 or tier 4. In our recent evaluation, we studied 132,000 variants classified as benign or likely benign in ClinVar. Specifically, we focused on benign likely benign variants within the TSO500 genes, ensuring their confirmation as benign based on rigorous criteria like practice guidelines, expert panels, and validation from reputable submitters. Nearly all these variants, about 99.9%, were correctly identified as clinically non-relevant. For the remaining 65 variants, 64 were deemed non-relevant. With their clinical insignificance supported by published studies or due to insufficient evidence opposing their benign nature. There was an intriguing exception involving a likely benign P10 variant in ClinVar, which the KB flagged as clinically relevant. This classification was based on its proximity to a spliced donor site. The variant involves a GGTA repeat sequence crossing the boundary between exon 4 and intron 4, causing uncertainty about its impact on splicing. This unique case prompted us to reassess how we handle P10 splice site variants in our KB, recognizing the need for a nuanced approach in representing these variants.
Gargi, thanks. Great. So to wrap up here before we leave some time for questions, I wanted to talk some about the clinical genomic workspace, which is the platform on which our lab customers can experience our knowledge base, can extract from that the information they need to rapidly interpret and sign out reports. We developed several strategies to make this a faster, easier process, a more intuitive process for users, and help them to focus their attention on new and changed evidence, and benefit from their past experience in our system signing out cases. So the first point is that we have built a memory system that not only remembers how you tiered a variant in a past case for this similar disease context, and not only remembers its interpretation, which we've done for some time, but also remembers the discrete decision that you made about whether to report that discrete drug or a prognostic or diagnostic association on the report, and what order you chose to report these associations in for that biomarker. Likewise, it's going to the far right. We've this memory allows us to take a snapshot of what evidence was available to you as a user the previous time that you encountered the biomarker, so that the next time you see this recurrent change, we can highlight for you how the evidence has evolved, what's new, how have assertions changed since then. You can focus your attention on those and make decisions about how to change your interpretation accordingly if appropriate. And then finally, we have a robust way to automatically draft interpretations, including for TMB and MSI, so that you have some great starting material that you can use straight up or edit before you sign out the case. So a few screenshots to illustrate these points. Here we go. So first, here's a view of a BRAF V600E alteration in papillary thyroid carcinoma, and you can see that the assertions that we have curated in our knowledge base that we've aggregated across sources are displayed here. There's a number of drug approval and a number of drug treatment recommendations from the NCCN, and a few pieces of emerging evidence that we've curated from PubMed. You can see that some of these are flagged or toggled as being in the report and some out. You make a decision here and about what you want to include in the report and what disease term you wish to associate with that. If you want to summarize some of the terms from our ontology into a shorter thing, you can do that. And you can also change the order of the associations as they will appear in the report. Importantly, when you set up a panel in our system, you can specify which sources you want to use for automated tiering and to appear on the report by default. So for instance, US users may choose not to leverage EMA and ESMO specific evidence that, assertions that are made only by those European sources. They're always still transparently displayed here for you to look at, but they won't automatically be in the report or be used for tiering if you choose not to use them. You could always decide to put them on after all on a case-by-case basis. Now, bubbling back up to the level of all of the key identified findings in a case, we've listed them here in tier order. They're automatically tiered based on the AMP-ASCO criteria as we've described a few minutes ago. And this includes multivariant findings or co-mutations that are being interpreted here by the system as a distinct entity from the individual alterations of the HER2 gain and the MET splice variant. Those are listed here individually, but they're also listed together and are tiered at 2C based on either emerging evidence or maybe trial associations. You can also see that each of these has a colored tag that indicates whether or not the alteration has been seen previously by you or by your institution in this disease context. If you have, and there's no change to the evidence, it's tagged as familiar. And if you have previously encountered the biomarker, but now there's change or new evidence, then it's flagged as an update. And so you could focus your attention on those alterations that are novel in your experience in this disease context or for which they're updates. So here we're looking at the same example alteration, in fact, and this NCCN and FDA assertion for dabrafenib plus trametinib in the context of solid tumor, you can see has an indication here, a little orange triangle with an exclamation point that indicates there's a change to the assertion. And you can see blue stars too, which indicates, hey, since you last saw this alteration, these are new assertions that have been added to the knowledge base. So you click on this one here that we described for dabrafenib and trametinib, and you can see that, hey, NCCN now supports this assertion. That's the change, and you can make a decision on if you care about that change or not. You might want to change your reporting decision based on that. So in this way, you need not review, especially for these recurrent alterations that are well studied and have lots of either recommendations or drug approvals. You can do the work of sifting through that evidence one time in a disease context and make your decisions about what to report. And then subsequently, you can just focus on how the evidence has changed and build off of your past experience. Likewise, the discrete decision here for this TP53 truncating alteration in NSCLC, the discrete decision of whether to report or not is saved. So if you toggle this thing off, it'll say, hey, we're saving that decision so that next time you see this alteration in non-small cell lung cancer, we'll have it out of the report by default. Now, there are cases when you might choose not to report for a given patient. Say there's a drug association, but that drug has been tried on that patient and you don't think it's useful to put it in the report. You can turn that off, that assertion off. It is not in the report. And you can say, hey, don't save that decision. You can click on that little bookmark icon and you can tell the system, please next time bring that back because I generally do want to report it. So you have flexibility there on how the memory works. And I'll just reiterate here that the combination of alterations is, a critical thing to consider. And because the system is going to automatically flag combinations that have some medical meaning that is distinct from the individual alterations, you can really find out some important pieces of information, such as in this non-small cell lung cancer, a KIF5B RET fusion is likely to benefit from selpercatinib treatment. But according to emerging evidence from PubMed, in combination with a MET amplification, it may not work after all. So you can evaluate the evidence from the individual variant and from the combined variants as a distinct biomarker or a distinct finding, and determine from there whether you want to report one or both of those pieces of evidence or both of those findings with their associated evidence. And then you can always clarify and summarize the overall findings of the case in the executive summary to describe, this combination as being an important point. And then, based on what you've selected as discrete assertions to report on your report, the interpretation will be drafted accordingly. So on the right is the system-generated interpretation based on the decisions you've made on discrete assertions, and kind of broken down by therapeutic, prognostic, diagnostic, either within your patient's tumor type or within another tumor type. And then you can kind of promote that to the interpretation text here. You can edit freely to finalize for that biomarker. Just a quick point about clinical trials. Here, of course, we have matched, again, based on disease and on biomarkers. So a trial will only appear here if your patient is eligible, at least with respect to the disease and biomarker criteria. And they've been ranked by phase and by distance. So at time of accessioning, you can specify the country and postal code of your patient or your lab if that's the best localization information you have, and the system will then rank for those trials that are most promising in terms of advanced phase and are within a reasonable distance or closest to your patient. You can always make decisions to change which of those trials you want to report. In this case, there's a whole bunch of additional ones that match but aren't in the report by default, aren't the top 5 matches. And you can configure whether you want 5 or 2 or whatever as the default number that are in the report at the point that you first come into the case. You can also specify and configure which clinical trial registries you want to be considered. If you're a US-based lab and don't want EU-CTR trials to be considered, you can specify that. Of course, they would also be ranked lower on average because the locations will be in Europe for those trials, so they would be far from your patient, presumably. And then I failed to remember this bit, that we have built based on the Belgian Compermed group's recommendation, a biological relevance or biological classification automated determination. So especially for those alterations that on AMP ASCO CAP scheme are VUS, you can come into this tab and view a series of of rules that we've run on the variant to determine whether it's likely benign based on being common in the population, or our known loss of function alteration in a known tumor suppressor oncogene, or if those fail, in this case it was clearly a consensus pathogenic variant based on Compermed's list. But we can then calculate a parameter score based on how frequently it occurs in cosmic, the in silico predictions, and ClinVar evidence. And so based on this, you can get additional information about possible biological relevance in a disease agnostic fashion of a variant that may not have clear actionability. And of course, all of this is in the direction of generating a concise report that gets the ordering physician the information they need on what you found from that tumor genome that informs care of that patient. You have lots of options in our system as a laboratory to design this report with your branding and colors, to, as I mentioned, specify or rather configure which sources you want to consider for reporting, and how you organize this report, what sections you include, etc. So that's a little whirlwind of some of the capabilities in the system to leverage the knowledge base information. That's all I had. I meant to put my email address on here, but I can put that in the chat, and you're free to reach out to me or to Gargi to ask further questions. But we have some time, and be happy to entertain questions. Thank you for your attention.
Sure, thank you. That was really great. Just a couple things before I open it up to everybody on the call. So Velsera is a commercial product. Is there any portion of the product that is publicly available or might be publicly available at some point in time?
Yeah, great question. So we've, as we were developing some of these processes some years ago, we looked at FDA's guidelines for recognizing genetic variant databases, and we've followed a lot of those processes. But at this time, this is part of a commercial product, and we don't intend to open source the knowledge base or our processes. So
Sure.
it's still a proprietary product. Unless I'm forgetting something, Gargi, we don't currently have anything that's outside of our commercial offerings.
Mhm. Oh, that's totally fine. I just wanted to be transparent with those on the call,
Thank you.
because yeah, this is a really great presentation, and just want to make sure everybody's in the know about that. Speaking of FDA, kind of the hot topic in lab medicine today is like additional oversight. So you had mentioned early in the presentation that you help labs with their validation of different assays as part of Velsera. How do you see that changing with additional oversight from FDA?
Yeah, that's a key question. I think that I won't say that we are helping labs meet the very bar that FDA will, put forward under their proposed rule. But we are well on our way. We have helped numerous labs meet the necessary requirements for validation, analytical and clinical validation of their assays under CAP, right? And so I think we're well positioned to step that up as additional support. But yeah, it's going to be additional work. I share many of the concerns that the AMP and others have put in their comments. But I believe that between our own experience working with FDA for medical devices and our validation experience, that we'll be able to support labs through that if they're game. Yeah, there's going to be more overhead, but I think that philosophically it is not that different from what we do today to support labs with validations.
Great. Thank you for that. Does anybody else have any questions for Andy or Gargi?
Sure. This is Deb Ritter from Baylor College of Medicine. I just had a question on, like, what do you do with literature that you might toss out, or do you ever do that? Like, so if you go through and, you find that a study you were relying on maybe wasn't up to standards or anything like that, what do you do with that pile of papers or sources that isn't used?
Yeah, so, we're pulling in papers weekly, right? And so there will be times when we have evidence or we've curated papers, but that particular claim from that paper isn't supported by others, and there's no consensus, right? And so it doesn't ultimately make it through the aggregation phase, and it's not represented in that final knowledge base. But it's still sitting out there waiting for additional support, okay? And so then as another paper comes along, the two together may be enough to meet ASCO-CAP Tier 2C, for instance. Now, should additional evidence come out that is in opposition to that paper, and then again, we're going to reassess the consensus algorithm as we go, as we add more papers. And so that could change. Something could have consensus, and then eventually there's too much conflicting information and it drops out, and there's no longer a consensus interpretation. If there's like an erratum or a retraction published, we would catch that and likewise recalculate is the overall evidence enough to build consensus. Does that answer the question? I mean, we're reevaluating on a constant basis, so nothing that goes in is like there permanently. It's going to be based on additional evidence that may come out.
Great. Yeah, I mean, it does. I realize the reevaluation. I was just kind of wondering, I mean, sometimes, we're so focused on like the data that's really good, or the papers that are really good, that like understanding maybe like what's not, like which papers are really bad, and how can we learn from like what's the bad data in papers, or like what papers like don't have supportive evidence, et cetera. And then kind of just understanding how you do that, like you said, so you're searching for erratum papers or something like that to go back, like what would kind of trigger that search to go back and say like, oh, after we've seen these, I don't know, phase 3 clinical trials, we actually have a lot of data in phase 4 or something. I don't even have a great example in my head, but just was thinking about it. So thank you for the answer.
Yeah, sure. I mean, I think an erratum would come through as a new, hit in our search, so we would pick it up that way. We don't make subjective decisions about whether a paper is good or bad, but we do have, and Gargi, you may remember this, there are certain different thresholds for consensus. If there's evidence that is, not clinically significant, like we don't ignore it, but we treat it somewhat differently. And there may be some other parameters that I'm forgetting that help us kind of weed out some questionable results. Do you have anything to add to that?
Yeah, the mutation status and clinically the outcomes of the studies. So the study outcomes and whether the data is significant or not. So we assess all of these parameters and check whether the variant is associated with the clinically relevant outcome and it is significant. So these all criteria actually match, and then we qualify that biomarker, variant, and assertion. Like together, we qualify it for evaluating with other biomarker disease combinations and assertions. We catch it from different PubMed articles, and then we evaluate.
There's a rather involved decision tree that's at play here. So thanks, Gargi.
Okay.
Thanks, Deb, for the question.
Great. So I have one more question for you. So you had mentioned the different classification schemas that are supported and recognized by Velsera, including ComparMed, which is in Belgium, versus ESMO versus ACMG, AMP-ASCO-CAP. Do you find that your clients who are not from Belgium or Europe will look at the classification from those other schemas and on tricky variants and help tip the scale one way or another?
Yeah, I can't say that I have a lot of intel on what, say, US customers are going to do with ComparMed. You know, it's I expect that it is useful adjunct information. I don't expect that US labs are going to use ComparMed for reporting, right? Like parallel to AMP-ASCO-CAP. You know, there's also, of course, the VICC oncogenicity scheme, which we have not built, but you might have seen in that interface as a drop-down for ComparMed. So you can, you know, we have plans to be able to support additional schemes. And we haven't, you know, had people are not clamoring in the clinical space for, you know, for that. I think ComparMed was a something that 2 or so years ago, there were, you know, customers in Belgium who, you know, that was a requirement. And so we built that out. But yeah, I think that it's useful to dig in and, you know, have a synthesized view of some of the evidence from, you know, Cosmic and ClinVar and such. And, you know, it probably doesn't necessarily change the actionability of an alteration, but it may help somebody decide to put something into 2D and talk about it.
Right.
That's my sense of it.
Yeah. And I think that I don't know, I would probably just be curious if I was working up something and it was like a VUS and I was kind of on the edge between calling it like a tier 3 versus a 2D. And maybe that would help tip the scales a little bit. And then you answered my question about oncogenicity. So great. Thank you. Are there any other questions?
Since we have just a couple minutes, I did have a question on the historic version access and how does the user experience that? So is it if they want to go back like 2 years prior or something based on something that they've done? I mean, I haven't used the platform, but if they want to go back to look at that, what is it that they do? They use a version and go back to that exact view that they had before, or how does it practically work?
A version of the knowledge base, is that what you're referring to?
Yeah. You were saying that, you know, if they made a decision based on the knowledge presented at the time, they can go back and access that and figure out kind of what changed.
Oh, okay, got it. Sorry. So the feature I described was of being able to spot, right, what has changed, like what's new or retired or you know, changed. And so you can infer from that what the state was last time. We don't have a specific way for you to go review the I mean, you can look at the old case, right? You can find the case that you reviewed previously and you can look at the evidence, but you can infer it like from point A to point B based on what we indicate in the system, right? Okay, if that's retired and that's added, and then I can tell what I had last time. And,
Got it.
and
Okay.
all of those assertions will be, you know, if you click on them, they'll say, hey, there's been no change since last time you saw this. Or, you know, we updated the version of the NCCN practice guideline, but otherwise it's the same claim. It just, you know, it's now still current, right? That's
Okay.
kind of the information you get.
Okay.
Great, thank you. Are there any other questions? We just have a few minutes left in the hour. All right. Great. Well, thank you again so much. I think we'll give everybody 2 minutes back. But what
Deal.
a great presentation. Thank you for being with us today and sharing what you do with our group. So looking forward to interacting with you two more in the future through VICC.
Thanks for hosting us, Beth and Wes, and all. Appreciate it.
You bet.
Thank
Have
you.
a
Have a good
great
day.
week, everyone.
Good night.
Yeah.
Bye-bye.
Bye.